But if you do manage, and obviously we do, manage to package that 3 meters of DNA into the nucleus, it's going to be hard to find the genes that you want to use. And that's where these epigenetic marks become important. We want to mark the regions that we want to use, and we want to mark the regions that we don't need to use so that they will be silent. And since the title of my talk is about inactivation, obviously I'm going to be focusing on this silencing aspect. But I want to emphasize that silencing is incredibly important because there's actually much more DNA in a human cell that we want to keep silent than that we're going to be using. And so some more scary math. If we actually look at the human genome and we say back in that central dogma I said there were DNA going to RNA going to protein, how much of that DNA is actually telling the cell what proteins to make? And the answer is something around 1% of it. So the DNA that actually codes for a protein is about 1.5% of our DNA. One way as biologists that we say what's important DNA for a cell is to say, well, do we also find it in cells of other animals or in fact other plants and yeast, and how conserved is that DNA? And the very, very basic important structures like those histone proteins, they're conserved right back to yeast. But if we look at even the well-conserved protein, we're still well under 5% of our DNA. In actual fact, over half of our DNA we can still identify as being remnants of invading retroviruses that popped into our genome sometime in ancient history. And so we need to keep that DNA silent. So we need not only a mechanism of identifying the important bits, but we need a mechanism of silencing all the rest of it. So when we study genetics, we're studying the changes that occur to the actual DNA. But what we're talking about now is epigenetics. The DNA stays the same, but the outcome is different. The cell does something different. The organism does something different with that same DNA.
So what is epigenetics? So it's important to define things before you start just talking about them. If you don't know what something is, you got to stop and define it. So epigenetics, as an organism grows and develops, carefully orchestrated, no kidding, chemical reactions activate and deactivate parts of the genome at strategic times and in specific locations. It's an amazing thing. We're constantly trying to figure it out. And epigenetics is a study of these chemical reactions and the factors that influence them. So let's talk about an epigenetic example, epigenetics versus genetic cause of disease, because we do have genetic causes of disease, but for the most part, I think we have epigenetic causes of disease. So if you look at Crohn's disease and ulcerative colitis, if you look at the genetics that are associated with these, there's about 70 genes that are associated with ulcerative colitis and Crohn's, 70. So that's broad, that's extremely broad. So if there's that many genes that are related to these two conditions, for a clinician standpoint, that tells you that it's epigenetically controlled. There's not one gene controlling the outcome of this. If you look at celiac disease, there's a single HLA locus, a single gene that's highly, highly related to celiac. If you have twins and they both have the celiac gene, more than likely they're going to have celiac disease. So let's focus on main epigenetic modulator, which is methylation. So the addition of a single carbon group with 3 hydrogens, that's what methylation is. And it's not only about folate. There's many methyl donors. There's B12, there's choline, there's glycine. These things are all methyl donors. But the biggest one that we talk about is folate, and that's where MTHFR comes in. And if we look at these two compounds as an example, uracil is an RNA base. We all know that. but we don't know what methyluracil is. Methyluracil is thymine. So in order to convert uracil into thymine, it requires methylation. If you have inadequate methylation in your patients or yourself, you're going to be incorporating a uracil base into your DNA. Why is that a big deal? Well, it's because uracil is not supposed to be in your DNA. It's an RNA base. But thank goodness we have repair mechanisms in our DNA. The problem is every time you're repairing your DNA, you're splicing it. You're cutting out that uracil, and you need to be putting in a thymine. Every time you do that, it causes fragility in the DNA. Over time, it's going to be more and more fragile, and it's going to be hard to read. So functions of methylation, there's a ton of them. And since if you look at all the different functions of methylation, it shows you the importance of it. So methylation, for the most part, will turn genes off. So we have cells all throughout our body, and they all house the same code. So we have genes in our eyes, which allows us to see and what have you, and we have the same genes in our eyes, in our arms, and our legs, and our feet, right? But they're turned off. They're not expressing. We don't want to have eyeballs in our arms, so they're turned off. They're methylated, for the most part. So that's what methylation does, and it also biotransforms, gets rid of chemicals that are in the environment and also inside of us that we make on our own, and we make neurotransmitters from it, and we also get rid of those neurotransmitters. So you're building neurotransmitters and eliminating with methylation, both. So it makes it very tricky as a clinician to support your patients with methylation because you're building neurotransmitters, eliminating them, and you have to know what the symptoms are. And history is very important because labs for neurotransmitters are very tricky.
And he concluded that of the 88 foods they tested, almost half had blood group specificity. And if there's not direct blood group specificity, very often there's secondary blood group specificity. In other words, if the lectin is not specific for the blood group sugar that identifies the ABO blood type, it can often be specific for secondary sugars that are also associated with that blood type as well. An example of that is a type O antigen, which is not a true antigen, as you probably remember from hematology. But the type O antigen is a terminal sugar called fucose. So lectins that would be specific for fucose would have a tendency more to interfere with that blood group. But also, too, type O manufactures higher levels of a secondary sugar called N-acetylglucosamine. And so it tends to be more sensitive to those types of lectins, even though they're not directly related to the actual blood group expression. The single most important lectin that reacts with N-acetylglucosamine is the lectin from wheat germ agglutinin. So we'll go through this later in terms of significance. Here's a kind of a simple list of foods with known dietary lectins. I mean, these are the most common foods in the diet. They contain lectins that have been identified and been identified in terms of binding proclivities. If I had time, I would give you a lecture on lectins because it's a fascinating topic. And it's a direct relationship between a predictable component of a person's digestive tract and the direct consequences of foods they eat. But as you can see here, these are very commonplace foods that contain considerable amounts of immunoreactive molecules. And they're not necessarily going to react in anything that could be necessarily picked up on an allergy test or anything like that, but they're probably more likely to cause intolerance-type reactions. And they result in a whole series of secondary effects that have to do, for instance, with increasing what's called endocytosis, which is the ability of the digestive tract to become more permeable. The standard reaction, of course, is agglutination. And we see here on one side a smear of red blood cells that are non-agglutinated and a smear of red blood cells that are agglutinated. Interestingly enough, the actual action of the opposing blood group antibodies, in other words, when you get a transfusion of the wrong blood type, that's the same exact reaction because the opposing blood group antibodies are known as isohemagglutinins, right? Anti-A or anti-B. And they're very large IgM class molecules that are capable of agglutinating themselves. So the whole agglutination phenomenon and its blood group specificity even extends into the relationship and the interrelationships between blood groups in addition to the relationships between individual blood groups and their diet. Again, I could give you we could spend a lot of time talking about lectins with regard to many, many things having to do with chronic immunology type problems, but I think I want to focus on, at least in this lecture, was just simply gut permeability.